Hello, everyone, and welcome to today's webinar, Utilization of Optical Genome Mapping in Detection and Characterization of Rare Genetic Markers in Pediatric Leukemias. I am Alexis Corrales of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Genomics. To learn more about our sponsor, please visit them at bionanogenomics.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well, for example, if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Dr. Gordana Raca, Director, Cytogenetics Laboratory, Department of Pathology and Laboratory Medicine at Children's Hospital Los Angeles, and Associate Professor of Clinical Pathology at Keck School of Medicine at USC. Dr. Raca, you may now begin your presentation. Welcome.
Thank you very much, Alexis. I would first like to thank LabRoots and Bionano for the opportunity to share our experience with optical genome mapping. And I also want to thank all the attendees who decided to join us today for this presentation. So I will be telling you today about our experience with using optical genome mapping to detect and characterize rare genetic markers in pediatric leukemias. This is the outline for today's talk. I will start by briefly describing principle of optical genome mapping, and then I will discuss two studies. One was focusing on detection of rare genetic drivers in pediatric leukemias, and in the other study, we used advantages of optical genome mapping for characterization of one unusual structural abnormality in B lymphoblastic leukemia, known as PAX5 intergenic tandem amplification or tandem multiplication. So what is the principle of optical genome mapping? This slide is showing the workflow and principle of this technology. Starting from the top left corner, you first isolate high molecular weight DNA from fresh sample. And the first step is to label DNA at short sequences using fluorescent markers. Then the molecules are loaded on the instrument, which will unbind and linearize those DNA molecules so that labeling pattern can be visualized and imaged. In the next step, you can use the mapping patterns to assemble contigs, and then patterns can be compared between your sample of interest and reference. That allows you to detect all the places in the genome that you're testing where labeling pattern differs from the reference, and those changes are typically due to different types of structural variants and either copy number changes or balance rearrangements. So with this approach, you can detect a variety of structural variants in your genome of interest as compared to the reference. So on the next slide, we see different types of structural variation that can be characterized and detected by optical genome mapping. On the left, you see how optical genome mapping allows to detect copy number changes. And by comparing your samples in green and the reference, you can see deletions and duplications. You can also detect balance rearrangements, translocations, and inversions, for example. And then in the middle, you see how optical genome mapping can be used to characterize repetitive sequences, which is very difficult to do by other molecular technologies. And so you see examples on this slide of using optical genome mapping to characterize repeat array expansions or tandem duplications. And finally, by combining information from mapping patterns with information of coverage and number of molecules coming from different regions of the genome, you can get very nice genome-wide copy number profile and assessment similar to what you would get from chromosomal microarrays. So in summary, you can detect both balanced and unbalanced rearrangements, like you can from karyotype analysis, but with higher precision and resolution. And you can get copy number profiles similarly to what you can get from chromosomal microarrays. So now I would like to move on and describe our use of optical genome mapping in detection of rare genetic drivers in pediatric leukemias, which were missed by using our routine genetic testing methodologies. But before I describe our study, I want to give you some context and explain why we focused on acute testing and studying acute leukemias. Acute leukemias are a common tumor type in children, accounting for more than 25% of all cancer diagnoses in individuals younger than 20 years of age. And they still remain an important medical problem because they're still the second leading cause of cancer death in children, in spite of all the advances in treatment over the years. And importantly, genetic abnormalities have very significant role in risk stratification and choosing right treatment for patients with acute leukemias. But we are reaching the point where our traditional methods of testing karyotype and FISH are not efficient in detecting all genetic subtypes that are clinically significant in this disease. So just to briefly explain, why is karyotype and FISH not sufficient anymore for our clinical workup of acute leukemias in children? If we take example of BLL, about 15 to 20 years ago, we only knew a handful of cytogenetic subtypes, and maybe about 10 different cytogenetic subtypes that could be picked up and characterized by chromosome analysis. And in more than 25% of the cases, the underlying genetic driver remained unknown simply because karyotype could not pick up those abnormalities. But with availability of advanced genomic technologies like genome sequencing, exome sequencing, expression, clustering, RNA-seq, we now recognize almost 30 different genetic subtypes of BLL. And many of these new genetic subtypes are already clinically significant. We know their prognostic and therapeutic associations. But those are exactly the ones that historically we were not able to pick up by karyotype. And that represented that 25% we were not able to characterize. So nowadays, we actually can detect the genetics drivers in almost 95% of all the BLL cases, but those novel genetic subtypes need other genetic testing methodologies to be efficiently identified. In terms of testing challenges in pediatric acute myeloid leukemia, recent genomic studies have shown that the genetic profile in pediatric cases is completely different than in adults, very different types and frequency of abnormalities than what you see in an adult. And since most of the testing is focusing on adult cases, the assays that are easily available for either fusion detection or detection of sequence variants are tailored towards adult cases and do not work well for pediatric AML. In addition, in pediatric cases, you have very high heterogeneity of genetic drivers, and they tend to be fusions that are individually very rare, but collectively fusions are the key drivers in pediatric AML. And they arise from structural rearrangements, which are challenging to detect by existing methodologies with high resolution. And also because of high heterogeneity, targeted assays are not working very well. So you need methodologies that will allow for efficient genome-wide detection of structural changes. And at the same time, while there are all these challenges, the number of markers that are having prognostic significance is constantly increasing. So we want to be more and more efficient in detecting them at the time of diagnosis. So to address these new needs in genetic testing for pediatric leukemias, at our laboratory at CHLA, we implemented multimodal testing that, in addition to routinely using karyotype and extensive FISH panels, also uses routinely chromosomal microarrays and our custom next-generation sequencing panel that we call OncoKits, which was especially designed for pediatric cancers, including pediatric hematologic malignancies. However, even when we apply all these testing methodologies, our recent retrospective studies show that in BLL cases, we still have 15% of cases where underlying genetic marker remains unknown. And then in AML, we have about 5% of the cases where, with combination of our current testing methodologies, we still cannot identify the key genetic driver. So that prompted us to continue looking for more comprehensive and novel methods to do our diagnostic testing for leukemias. We are in the process of validating RNA-seq for fusion detection, and we are also validating exome sequencing for DNA sequence variants. However, we are constantly looking for other options for doing more efficient clinical testing, because the goal is not only to be comprehensive, but also to be very efficient and to have good turnaround times as well as reasonable cost of our testing. So that got us interested into optical genome mapping, and what in particularly sparked our interest were results of this multi-center study, which looked exactly in hematologic malignancies and utility of optical genome mapping and characterizing previously detected and novel structural variants. So in this study, they were able to show that optical genome mapping detected copy number abnormalities and structural variants that were previously detected by karyotype and FISH, but also detected additional changes that were missed by traditional methods in 11% of the cases. And it refined structural variants that were detected by karyotype but could not be accurately characterized in additional 24% of the cases. So it certainly looked like a very promising technology, especially seemed to work very well in hematologic malignancies, and we wanted to test it ourselves and see if it can help us in some of our unknown and difficult cases. So we did a small feasibility study to determine if optical genome mapping can detect clinically significant genetic drivers in pediatric leukemias, which were missed by comprehensive molecular evaluation in our laboratory. So we were able to find 9 cases which were previously tested and remained unknown, for which we had materials saved to do isolation of high molecular weight DNA. Those were 8 BLL cases, and so far, unfortunately, only 1 AML case. What was striking for us in these BLL cases is that out of 8 cases, 6 were classified based on clinical presentation as high risk, which showed us that in these cases with unknown genetics after our comprehensive evaluation, we have enrichment for high-risk cases. And I think that that stresses the importance of having good methods to and more efficient methods to tackle these very difficult cases that remained unknown, since those are the high-risk ones where you, from diagnosis, want to know as much information and choose the right approach. So I will dive immediately into our results with optical genome mapping. When we look at our 8 BLL cases, in 2 out of 8 cases, optical genome mapping detected previously reported and known driver fusions, which remained unknown after our comprehensive analysis. So I will describe these cases in detail. This was the first case. It was initially diagnosed as standard risk BLL, but unexpectedly it had poor response to treatment. So the patient had residual disease at the end of induction and had to be further treated more aggressively. And our genetic testing started by karyotype, which did detect 2 rearrangements. There seemed to be translocation between chromosomes 2 and 5, as well as inversion on chromosome 14. But at the time of initial diagnosis, we really did not understand the significance of these rearrangements. By chromosomal microarray testing, we did see a deletion of Icarus gene, which is known as poor prognostic marker in BLL. So that did make us concerned about this case, that it might not be just a simple standard risk BLL, but we didn't really know more specifically what were important genetic drivers in this case. So when we got optical genome mapping results back, it confirmed the presence of the same abnormalities that we saw by karyotype. We were reassured that it also showed presence of translocation 2.5 and inversion 14, shown on the left in the circus plot. But more interestingly and importantly, there were very important genes at the breakpoint for this translocation 2.5. On chromosome 2, it was SPT-BN1 gene, and then on chromosome 5, PDGFR beta gene. And we got excited because fusions that involve PDGFR beta gene are known to be associated with a high-risk subtype of BLL known as Philadelphia-like or PH-like BLL. And also, even more significantly, they seem to indicate response of leukemia to tyrosine kinase inhibitors. So you can target cases with PDGFR beta fusion with existing tyrosine kinase inhibitors. So we obviously wanted to confirm that the fusion was real. We have FISH probe for PDGFR beta, which clearly showed that there was PDGFR beta rearrangement. And since we are validating RNA-seq for fusion detection, we were able to run the sample by RNA-seq, and that also confirmed the presence of the fusion. What is the significance of this finding? As I mentioned, it's a fusion associated with PH-like BLL, which is high risk, so there is prognostic significance. There is treatment significance because these fusions respond to targeting by tyrosine kinase inhibitors. And then for us, the important question was, why did we miss that at initial diagnosis? And we didn't think when we saw karyotype findings about this particular fusion because fusion with this particular partner, SPT-BN1, has not been seen in BLL before. It has been described only in few cases of another hematologic malignancy, myeloproliferative neoplasm with eosinophilia and PDGFR beta rearrangements. And this is a dull disease that we didn't expect to, so we did not expect this fusion in pediatric BLL. And that also explains why this fusion was not part of our design for our OncoKids NGS panel. We also did not expect PH-like BLL subtype in patients who presented clinically as standard risk, although there are some rare cases of standard risk patients who end up being PH-like BLL positive. But with optical genome mapping, we were able to pick up this abnormality, and as I mentioned, there are some both prognostic and treatment implications. The second case where optical genome mapping detected previously published and described fusion in BLL, and there was a patient who also was diagnosed initially as standard risk, but he had even worse response to treatment. His disease was refractory, and he had to be enrolled in CAR-T trial to get him into remission. However, he relapsed very quickly after completion of treatment and was referred to CHLA for another clinical trial with CAR-T cells. We repeated his genetic testing when he was referred to CHLA, and karyotype came back as completely normal. The CMA also did not find any significant copy number abnormalities, and then OncoKids only found NRAS hotspot mutation and some variants of unknown significance, but nothing that would tell us what is the key genetic driver for this leukemia. So to our surprise, optical genome mapping detected a variety of structural variants that we were completely unaware of, and the karyotype was normal. So that showed us that we probably did not get leukemia cells to grow in culture, because this patient leukemia actually had a very complex genome. And among all these structural variants detected by optical genome mapping, one really stood out, which was a three-way rearrangement involving chromosomes 1, 12, and 6. So try to highlight it in the circus plot on the left. But at the breakpoint on chromosomes 1 and 12 were some important genes. On chromosome 1, it was MEF2D, and then on 12, FOXJ2. So why is this important? MEF2D fusions with different partners are known to define a specific genetic subtype of BLL. And then FOXJ2, it has been described as one of the partners of MEF2D, but it's a very rare partner, only seen in maybe one case in one cohort study, again explaining why this was not a part of the design for our OncoKids panel and why we missed it by our comprehensive initial testing. We ran this sample on RNA-seq, so the fusion got confirmed, and we verified that it was real. But the clinical significance of this is that MEF2D fusions are known to be associated with very high-risk disease. And so if this was known in diagnosis, it would certainly help to risk stratify this patient appropriately, and retrospectively, it really well explains his very aggressive clinical course and poor response to treatment. So moving on, in addition to known previously reported fusions, in 2 out of our 8 cases, optical genome mapping detected novel candidate fusions that have not been described previously, but may represent novel discovery. So one of these cases was a high-risk BLL, where our previous testing detected some non-specific copy number changes by array, but there was nothing that really classifies as known genetic driver in BLL. So optical genome mapping in this case suggested the presence of a rearrangement between chromosomes 6 and X with 2 very interesting genes at the breakpoints. ATRX on X and BACH2 on chromosome 6. So based on optical genome mapping, it was expected that there will be BACH2 ATRX fusion present in this sample, and we ran this sample as well on RNA-seq and confirmed that the fusion is real. However, this fusion has not been described previously in BLL and may potentially represent new discovery. And we're excited about it and think of it as a very good candidate because both partner genes have been previously implicated in BLL. ATRX encodes epigenetic regulator and has been shown to be mutated in relapse cases of BLL. And then BACH2 is a transcriptional regulator that has important role in regulating T cell function and B cell maturation. So based on their known functions, both these genes are expected to be important in BLL, that they may be important in BLL pathogenesis. So we are now working on further proving the functional significance of this fusion. And then the second case where optical genome mapping detected novel candidate fusion was another high-risk patient with very aggressive clinical course who was referred to CHLA after relapse for CAR T therapy. And in this case, optical genome mapping detected multiple structural variants that were missed by karyotyping, including inversion on chromosome 9 with STRBP gene and JAK2 gene at the breakpoints. So optical genome mapping results suggested the presence of STRBP JAK2 fusion, and we were excited about this result because JAK2 fusions are known to be associated with pH like BLL. Again, STRBP has not been described previously as one of the partners, and for this fusion, we still don't have RNA-seq confirmation, but it's another very promising candidate for a novel fusion. So to quickly summarize our experience with BLL cases, previously reported clinically significant fusions were detected in 2 out of 8 cases. And novel candidate fusions were detected in 2 out of 8 cases. And then I didn't really specifically show this in detail, but in almost all the cases, 7 out of 8, and the optical genome mapping detected structural variants that were missed by previous karyotype analysis, and that we were unaware of based on other testing that we did in the lab. There was only one case where optical genome mapping did not detect structural variants novel structural variants that we were not aware of. So as I mentioned, so far we were only able to test one AML case, but the results were very interesting, so I really wanted to share this case with you. This was infant AML case. This was a baby girl who presented AML soon after birth. And she was diagnosed with AML with minimal differentiation, or M0 subtype based on FAB1 classification, which is a very rare subtype, morphologic subtype of the disease. And it's very little known about genetic basis of this particular subtype. Our initial karyotype analysis showed what looked to us like possible translocation between chromosomes 6 and 20, but the bending pattern didn't quite fit, so we indicated with a question mark that we were not absolutely sure about the nature of this rearrangement. And the other unusual thing was that this was present in all the cells, and so we were not even sure whether this might be a constitutional balance rearrangement in this patient, completely unrelated to her leukemia. And all other genetic testing that we did, including FISH and CMA and OncoKids panel testing, was essentially negative. So when we got optical genome mapping results back for this case, to our surprise, there was evidence of rearrangement that was three-way, that was more complex than we anticipated based on karyotype. So it was rearrangement that did include chromosomes 6 and 20, but it also included p-arm of the X chromosome. And on the right is are more detailed results of optical genome mapping, showing the scheme of what was the nature of this rearrangement based on optical genome mapping results. So there was translocation of XP material to chromosome 20 to the Q arm. And then chromosome 20 material was translocated to chromosome 6, the Q arm, and then material from chromosome 6 was translocated back to X chromosome to the P arm. But in addition to translocation, there was also a small inversion in the long arm of chromosome 6. So there was first inversion of a segment of 6Q, and then it got translocated to the XP. So, of course, we were very curious to see if we can verify this rearrangement by looking back at the karyotype. And now, when we knew exactly what to look for, we were actually able to appreciate that chromosome X was also abnormal. And then if you look at this partial karyotype on the right side of the slide, you can see that one of the X chromosomes has abnormal looking P arm because that's 6Q material translocated there. And XP material is on the long arm of chromosome 20, and then 20Q material is the long arm of chromosome 6. So now we understand why the bending pattern didn't quite fit for us initially, but we were not able, without knowing what to look for, to accurately characterize this rearrangement. So I think that this illustrates one of the strengths of optical genome mapping that was also described in that multi-center study, that it allows you to more accurately characterize abnormalities that you see by karyotype. What was also exciting to us for this case was that at the breakpoints of chromosome 6 and X, there were some very interesting genes. On chromosome 6, it was MYB, and known oncogene and gene which is important in hematopoietic tissues. And then on X chromosome, GATA1, a very important transcription factor in hematopoietic tissues. So OGM results suggested the presence of MYB-GATA1 fusion, but if you look at the bottom, how these genes are oriented on their corresponding chromosomes, and if you just have a simple translocation between chromosome 6 and X, you would end up in having these two genes in opposite orientations, and that would not result in the productive fusion. And so that explains why there was inversion of a segment of 6Q before the material was translocated to the X chromosome, because only that inversion allowed to create productive fusion between MYB and GATA1. And so now everything was starting to come together and to make sense if we assume that this rearrangement indeed results in MYB-GATA1 fusion. So we were excited to try to confirm this by RNA-seq, and RNA-seq data very clearly confirmed the presence of the fusion. It also showed the splice variants that corresponded to what was previously reported for this particular fusion. And so the MYB exons 6, 7, and 8 were involved with exon 5 of GATA1, and that's what has been described previously for this fusion. And so on the right in the middle, you see the scheme from one of the papers that describes this fusion in another AML case. And they also show a fusion between MYB exon 8 and GATA1 exon 5. And as I described previously, the fusion made perfect sense because both genes have very important roles in hematopoietic tissues. And MYB is known to be very highly expressed in early stages in non-differentiated cells, and then its expression goes down during differentiation. And in GATA1, it's opposite. It promotes differentiation, so its expression goes up during differentiation process. And interestingly, these two genes co-regulating each other. There are binding sites for GATA1 in MYB regulatory regions. So as GATA1 goes up, it binds to MYB regulatory regions and participates to downregulation of MYB. So the hypothesis for this fusion was that when the fusion disrupts GATA1, then there is no binding of GATA1 to MYB regulatory regions. There's no MYB downregulation, and that results in very high expression, not only of the fusion, but also non-translocated normal MYB allele, and that interferes with differentiation and drives leukemogenesis. And so the fusion is has been described previously and has some very interesting biology. However, what was known about this fusion was very different from what our case suggests. So we think that our case will provide some novel insights into biology of this particular fusion. And the fusion is exceedingly rare. It has been reported only in a handful of cases, and the biggest case cohort, if I can put it that way, describing this fusion, only had 4 cases. And interestingly, they were all infant cases, and all patients were male. So the authors hypothesized that this fusion might be restricted to male patients because they only have 1 X chromosome and 1 copy of GATA1, and there is no normal copy to compensate when you have GATA1 participating in the fusion, and that drives leukemogenesis. And the other interesting finding in this paper was that all patients had acute basophilic leukemia, which is a very rare subtype, morphologic subtype, but it's different from what we saw in our patient. So in contrast to this study, our patient was a female, and our patient did not have acute basophilic leukemia. She had leukemia with minimal differentiation. Finally, we do not really think that explanation why this would be restricted to male patients really holds, because although male patients have 1 X chromosome and 1 copy of GATA1, females also have 1 active copy of GATA1 since the 2nd X chromosome is inactive. And so there is biologically really no reason why this would be restricted to male patients, although one can maybe hypothesize that there could be some compensatory reactivation of GATA1 from inactive X chromosome in females. And so we really think that this is not going to be restricted just to male patients, and that this might be under-recognized abnormality, and because when you only study these cases by karyotype, and since the genes are in opposite orientation, simple translocation is not going to result in productive fusion. So all these cases will have more complex rearrangements, and there will be no consistency from case to case, and that prevents from recognizing this as a recurrent abnormality, and recognizing this as a more common type or abnormality in leukemia than what we think. So I think that this is a very interesting case that shows how optical genome mapping helped us not only to find diagnosis for our patient, but to get some very interesting insights into a rare genetic abnormality in AML. And then from clinical perspective, all previously published patients did really well, and they responded very well to therapy. They were in remission at time of publication, and so did our patient. He responded very well, and so we think that this fusion will be recognized as being associated with good prognosis. So now moving on, what were the other insights about optical genome mapping from our pilot study? Since we had chromosomal microarray results for all our cases, we were able to compare copy number profiles from chromosomal microarrays at the bottom and from optical genome mapping on the top. And you can probably appreciate that they're very comparable. So optical genome mapping allows you to get genome-wide copy number assessment, and that's very comparable to what you would get from chromosomal microarrays. As I mentioned earlier, in almost all our cases, optical genome mapping showed structural variants that we did not pick up by karyotype analysis. And so cytogenetics, more commonly than we realize, misses structural variants simply because those abnormal clones don't grow in culture. And so I think this illustrates advantage of optical genome mapping, that it does not require growing cells in culture, because that, as we've seen from our small pilot study, often leads to false negative results. And finally, what was very important to me as a cytogeneticist was that optical genome mapping gives you information about chromosomal mechanism, about the nature of chromosomal rearrangement that results in an abnormal fusion. When you detect abnormal fusions by methods like RNA-seq, you can see the presence of a fusion, but you really don't know what is the chromosomal rearrangement that resulted in the fusion. And with optical genome mapping, you do get that information. We were able to find fusions that resulted from complex three-way rearrangements and from inversions. And I think that that is also important in understanding mechanisms and understanding basis and pathogenesis of these important mutations in leukemia. So to summarize the first study, in the first part of the talk, in our unknown cases, optical genome mapping detected known clinically significant fusions, but also some novel candidate fusions, illustrating both clinical and research potential of this technology. It showed very nice concordance with chromosomal macroarrays in detection of copy number abnormalities genome-wide, and detected presence of structural variants in all the cases with normal karyotype and in almost all the cases that we included in our study. And finally, it allows to decipher chromosomal mechanisms that generate oncogenic drivers in contrast to RNA-seq and fusion detection from RNA. So now I would like to move on and describe how we use optical genome mapping to characterize rare abnormality in BLL that I refer to as intergenic tandem multiplication in PAX5. So in this second study, we tried to utilize another advantage or a strength of optical genome mapping, which is its ability to characterize repetitive sequences. As you know, characterizing repeats is very difficult by molecular methods. Karyotype has too low resolution for many types of repeats in the genome, and chromosomal macroarrays usually don't even have any probes for repetitive regions. And short-read sequencing also doesn't work very well because reads from repetitive regions cannot be mapped back to the genome. But optical genome mapping provides the advantage that you can directly visualize these repetitive regions in long DNA molecules, and you can really efficiently detect and study repeats. One well-known example are repeats associated with rare genetic disorders, facioscapulohumeral muscular dystrophy. So in this disease, disease is caused by decrease in the number of repeats and in the region of chromosome 4 by repeat contraction. And it's a very difficult region to study. Before optical genome mapping, the only way to detect this abnormality and make a diagnosis was southern blot. But optical genome mapping allows to very clearly visualize these contractions of repeat arrays. And so that now there are already clinical laboratories that have validated testing for this disorder using optical genome mapping. So in our next study, we use this ability of optical genome mapping to characterize repetitive regions and to get insight into this rare abnormality in BLL that we got interested in. So the way we got interested in this abnormality was starting from a specific case. We had a case of BLL standard risk where after all our testing, we really could not tell what was the main genetic driver. However, we really scrutinized the data, wanting to know what was the key finding in this leukemia. And we saw a very small copy number gain that overlapped with a part of the PAX5 gene. And PAX5 abnormalities are pretty common in BLL, so we first did not think much of it. But since there were no other genomic drivers in this particular case, we decided to look a little in more detail. So the duplications seem to involve 5' end of the gene, exons from 1 to 5. And then to our surprise, it actually turned out that this was, at the time, known recurrent abnormality in BLL. At the time, it was described and studied by 2 European groups who studied it by MLPA. And what they found was that this occurs in about 1 to 3% of BLL cases. That it typically involves exons 2 to 5 within PAX5, and so very similar to what we saw in our first case. By FISH, they verified that this was within PAX5, that extra copies were within the gene, that they are not inserted somewhere else in the genome. And then what was particularly interesting is that this did not occur in any cases that had other major subtype defining genetic drivers. And so it seemed mutually exclusive with other major drivers, and they made hypothesis that this abnormality probably defines a new genetic subgroup of BLL. A very interesting exception was P2R1 CRLF2 fusion, which was seen in subset of these cases. However, this fusion is tricky because it can be primary driver in CRLF2 positive PH like BLL, but it can also be secondary abnormality in a variety of different subtypes. So authors hypothesized that this could still be a secondary abnormality with PAX5 copy number gain being the primary. And they refer to this abnormality as PAX5 intergenic amplification, but I think that the number of extra copies does not quite reach the definition of amplification, so I mention it as intergenic multiplication. So then further insight into this abnormality came from studies in 2019 that defined a completely new subtype of BLL that was driven exactly by PAX5 abnormalities. So they referred to the subtype as PAX5 alt, and they show evidence that PAX5 changes are primary drivers in this subtype, although they are commonly also seen as secondary in other subtypes. And interestingly, they had a significant number of cases in the PAX5 alt cohort where they saw the same intergenic multiplication that we saw in our initial case. And so now this suggests that the abnormality is indeed associated with its own subtype, and that it probably is one of the abnormalities that define the PAX5 alt BLL. So we continue to be very interested with the thought that there are still many unanswered questions about this abnormality, and so we decided to determine its prevalence, its exact structure, and associated genetic changes in our cohort of BLL. Because we took advantage of the fact that we routinely do chromosomal microarray studies, so it was very easy to us to go back and retrospectively analyze all our cases and see how often are we detecting this abnormality. So in a cohort of more than 200 cases, we had 5 cases of this PAX5 abnormality for the prevalence about 2%. And then based on array data, it seemed to have pretty consistent size, about 25 kb, with only the first case that I described being a little larger. And it always seemed to involve exons 2 to 5, as previously described. And then by a chromosomal microarray, it was obvious that there was more than one extra copy. But based on CMA, it seemed that there are a total of 4 copies. But we were also aware that maybe CMA does not exactly accurately determine the number of copies. So it was very intriguing to us that you never seem to have just one extra copy. There always seems to be more than one extra. So we really wanted to verify what is the number of copies, what is exactly the structure of this abnormality, and what is the orientation of these extra copies of the duplication. And we thought that optical genome mapping could be exactly the right tool to answer our questions, because we expected that in long labeled molecules that you use in optical genome mapping, you will be able to directly visualize these extra copies and see exactly the number and the structure. So we were able to find residual material in 2 out of the 5 cases that were positive by CMA. And these are optical genome mapping results for the first of those 2 cases. And to our surprise, there seemed to be 4 extra copies for a total of 5, which is higher than what was suggested based on CMA. But we were very confident that this is the correct result, because optical genome mapping allows to also measure the size of this insertion, if we can call it that way. And then the size was also consistent with having 4 extra copies for the total of 5. And then in the second case, we actually had 5 extra copies for the total of 6 copies. Again, much higher number than what we thought based on CMA results, but this was also verified based on the size of the insertion. So this showed us that copy number was underestimated by CMA, and that really to get full information about the range of how many extra copies you have in this abnormality, it would be important to study additional cases by optical genome mapping. The other insight that we got from our studies about this abnormality was about its structure. So in our cases also involved typically exons 2 to 5. And when you look at the junction between these exons, it is predicted at mRNA level that it would be in frame. So this abnormality is not really predicted to disrupt the gene, but rather to result in extra copies of a certain region of the protein. And these exons exactly code for DNA binding paired domain of PAX5. So you would expect to get a protein that has extra copies of the DNA binding pair domain, and that obviously is expected to change downstream regulation of PAX5 target genes, because this is a transcription factor very important for B cell differentiation. What was also very interesting to us is that in our cases, we also did not have any key genetic drivers. All cases were negative for other genetic drivers that define subtype of BLL. So our results are also consistent with this being the primary subtype defining driver, likely associated with this PAX5 alt subtype of BLL. But we did have 2 cases with P2R8 CRLF2 fusion as described previously. So again, we are recapitulating this previously described association of this abnormality with P2R8 CRLF2 fusion. And we also detected some other genetic abnormalities that have previously described to be enriched in cases with this PAX5 intergenic tandem multiplication. So in the conclusions for the second study, we saw PAX5 intergenic tandem multiplication in 2% of our cases. It involved typically exons 2 to 5, and based on optical genome mapping, we now know that there was multiple extra copies. In our cases, we saw 4 or 5 extra copies in direct orientation. This is predicted to result in extra copies of the DNA binding region of PAX5. And it seems to act as primary driver because it seems mutually exclusive with other genetic drivers that define subtypes of BLL. But it shows this very interesting association and synergism with P2R8 CRLF2 fusion. And finally, we verified that optical genome mapping is indeed a very good method to study structure of this abnormality. However, there are still very interesting remaining questions when it comes to this PAX5 intergenic tandem multiplication. I think that it would be important to determine number of extra copies in additional changes and to see what is the range. It seems to be required that there is more than one extra copy for this to be oncogenic. However, we really would need to know what exactly is the range. Interesting question is, what is the mechanism of these duplications? Are they repeat mediated? Why they recurrently occur at the same region of the gene? How they lead to oncogenesis? What are the downstream effects on the PAX5 function and downstream PAX5 targets? And then it will be important to understand fully its association, possible association with PAX5 alt BLL. Is this really one of the genetic drivers associated with this particular subtype of the disease? And finally, another very intriguing question is to understand synergism with P2R8 CRLF2 fusion, which seems to very frequently be seen in these cases that have PAX5 intergenic tandem multiplication. So I really advocate that more cases of this abnormality should be studied by optical genome mapping or a method that allows to accurately characterize the structure of this abnormality. So to finalize, our overall conclusions from the two studies that we were able to conduct so far using optical genome mapping. In first study, we verified that it can detect previously undetected, known, and novel fusions in pediatric leukemias, while also detecting abnormalities that we previously detected by our routine testing, like karyotyping, FISH, and chromosomal macroarray studies. And then in the second study, we demonstrated that optical genome mapping allowed accurate characterization of PAX5 intergenic tandem multiplication, so it has advantage in studying repetitive regions. So with that, I would like to thank all the collaborators on these studies, and my colleagues in Hematology Laboratory in Center for Personalized Medicine at CHLA, and also members of the Gordana Genomics team who helped generously with running and analyzing our cases and interpreting the data. And of course, I want to thank all of you again for joining us for this presentation. Thank you.
Thank you, Dr. Raca, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we will answer as many questions as we have time for. So let's get started. Dr. Raca, our first question from the audience is going to be a two-part question. The first part is, based on what factors did you decide on selecting particular patients to test with the OGM on cytogenetically normal patients?
So our key criteria was whether we were able to identify a main genetic driver for that case. And so if we did all our testing in karyotype, FISH panel, chromosomal microarrays, and our OncoKIS panel testing, and we still could not detect the key genetic driver. And there are known drivers in leukemias that define subtypes of the disease. And so if we couldn't find one of those cases we consider unknown, unclassified in terms of their genetics. So we really focused on those. Those are the ones that are still obviously very challenging to us. And then if we also knew about the patient that had unusual clinical course, that disease did not respond well to treatment, that the patient relapsed, then that made it even more important to understand what was genetics of that particular leukemia, since there are many genetic abnormalities that are known to be associated with high risk and aggressive disease. So those are our criteria, but then there was also a practical criterion, whether we really had the material left and to do optical genome mapping, because these were all retrospective studies, and it was all based on cases that we tested previously in the lab, and sometimes we do have some residual material left, sometimes we don't. So there was that practical limitation.
Wonderful. And so the second part to this audience question is, would you see this technology in a routine diagnostic lab along with NGS?
I think it has really good potential to be used and to really be advantageous in clinical testing. There are some laboratories that are very quickly, very actively working on validating this for clinical use, because in the research setting it has shown advantages in detecting structural variants. It has many things that are attractive when you look at a test being used in a clinical setting, and I know that some labs are already working on validating it for clinical use. And there was recently a very nice symposium organized by Bionano with speakers from all over the world sharing their experience with optical genome mapping, and several laboratories described moving this towards clinical validation. And because, as I mentioned in my talk, it detects very well structural variants, both copy number changes and balanced structural rearrangements, which is what you can do by karyotype, but it detects that with better precision, with higher resolution. So it potentially could be a very good alternative to karyotype analysis and FISH testing. Specifically, we are interested in hematologic malignancies, so in that area, and we have now our own hands-on, firsthand experience on our own cases showing that it detects abnormalities that we missed by other technologies. So I certainly see potential, and I know of laboratories that are already validating it for clinical use.
Great, thank you so much. So back to another two-part question for you, Dr. Raca. When validating fusions using RNA-seq, was the sequence mapping done against a reference genome or a reference transcriptome? And why did you choose that reference type?
So it was mapped against reference transcriptome. I wish I could give you a lot of technical details, but I don't, because I'm not a bioinformatician by training. We have a bioinformatics team at CHLA, and that was helping us to develop and implement this test. So mapping is done against the transcriptome, but unfortunately I don't really we have a lot of technical details that I can share today. I will certainly be happy to provide those details in the future through email or other methods.
Great. Now, do you believe, I'm so sorry, here's our next question. So what CMA testing platform are you using?
Our laboratory uses Cytoscan HD arrays from Thermo Fisher, previously developed by Affymetrix. So that's what we use whenever we have fresh material, and both for our constitutional cases and for our oncology cases, when we get fresh material, which is most often the case for leukemias, and then for some subset of our solid tumors. But we did validate OncoScan platform as well for FFPE samples. We have that validated in the lab as well and use that when we get FFPE.
So we do have time for a couple more questions, but I do want to remind our audience, any questions that we aren't able to get to and those that come in during the on-demand period will be addressed by Dr. Raca via the email address that you provided at registration. So, Dr. Raca, do you think that there is a gender bias, for example, male predominance, for leukemia driven by the MYB-GATA1 fusion? And if yes, What is the possible mechanism?
Yeah, thank you for that question. I really thought that case with MYB-GATA1 fusion was quite fascinating. Since all other previous cases were indeed male, that might indicate that there could be potentially gender bias. And interestingly, it has been described for other genes that map on X chromosome, for other oncogene and tumor suppressor genes that map on X chromosome, that there seems to be, when they are key drivers, that they are more commonly seen in male patients. And so, although functionally females also have just one active copy of those genes, there still seems to be, in other cases as well, some male predominance when the gene is mapped on X. But what exactly would be the mechanism is a very, I think, interesting research question, and whether there is some, as I mentioned earlier, some type of compensatory reactivation of the inactive copy in females that makes those leukemias, those genes, makes it more difficult for these genes to be fully oncogenic and realize their oncogenic potential. So I think that that is very, very interesting research question that we might look into, or I'm hoping that some other groups will be interested in. But to answer the question directly, I think that there might be some male bias based on the fact that other reported cases were all in male patients.
Wonderful. So it looks like we have time for one more question. So Dr. Raca, first off, thank you for the fantastic webinar. Would you like to comment on the impact of the percentage of abnormal clone on the utilization of OGM?
Yeah, that's always a big concern in cancer testing. We want to be able to detect abnormalities that are present at a low level. And I know that there are studies out there that looked into sensitivity of optical genome mapping for abnormalities present at low levels. So it depends a little bit, similar to NGS, depends a little bit on how, if I can put it that way, depth of coverage or how many reads you're going to analyze. But I know that they have shown in previous studies that it can go down to 5% sensitivity. So it can detect abnormalities that are present as low as 5%. And then if you really push it and do a very high coverage, maybe even lower than that.
Wonderful. Thank you again, Dr. Raca, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Genomics, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us. Oh, excuse me, I've resaid that. And their interesting questions. Questions that we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information that you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. Stay healthy, stay safe, and until next time. Bye-bye, everyone.